Hello, my name is Emily Van Buren, and I am a PhD candidate in the Midlars Lab in the Department of Biology. Today, we'll be reviewing machine learning techniques that have provided insight into potential biomarkers for coral disease fate. So coral health has been at the forefront of coral reef research since the 70s, and the Caribbean is currently a hotspot due to the high prevalence of diseases within this area, two of which are recent tissue loss diseases called white plague and skittle D. Coral research has expanded into transcriptomic studies, and we've obtained a lot of information regarding innate immune system capabilities of corals and their responses to pathogen challenges. However, we're still missing key information regarding coral disease risks so that we can properly model disease dynamics within these coral ecosystems. One way we could potentially do this is biomarker research. It's thought that corals may produce biomarkers either through front-loaded genes or differentially expressed genes prior to a pathogen exposure, as these biomarkers have been found in a similar pattern in heat and bleaching tolerance studies. By using machine learning approaches that have been designed for cancer biomarker research, we can identify candidate biomarkers for either disease risk or disease resistance in corals. There are two disease experiments that have been done in the Midlars lab that we can use in order to answer this question and apply our machine learning techniques. Overall, we have stony coral tissue loss disease and white plague challenged experiments, and they are both designed the same way where they have genetic matching. This means that one coral colony was selected and then broken into two. It was either exposed to a white plague or skittle D disease coral, or it was kept as a control. This control allows us a genetic baseline prior to the pathogen challenge that we can use to identify those biomarkers. And they have the same disease prevalence in both studies, which is helpful to us. We use three different types of machine learning for this study: principal component analysis, independent principal component analysis, and partially scarce discriminant analysis. The PCA analysis that we provided here, we did two different ways of this. We had all the transcripts that were available, so the genes that were expressed within all the species, and we also had the shared genes within the four shared species. And this actually changed how the groupings occurred. So while they do group based off their host species, you also see grouping based off of the phylogeny when you have all of the gene counts available to you versus the shared gene counts actually follow that disease prevalence. So going forward with our next tube, we'll just be using the shared samples because it's going to follow that disease prevalence that we're interested in. The IPCA results, we are looking at the significant genes that are driving this genetic profile that we see on this two-dimensional space. So not only are we seeing still that grouping based off of the host, but we are seeing some separation based off of the disease fate outcome of its counterpart. So looking at the heat map of it, we can see how if these genes are either highly expressed or downregulated during prior to a disease fate, and seeing how it divides up. We're still seeing host obviously driving a lot of this, but we are seeing some different patterns based off the disease fate. This is an unsupervised approach, so it's not taking into account our disease fate. So we can use a supervised approach like PLSDA in order to discriminate between the outcomes. And so we can see that we do have some discrimination that occurs. There is some overlap, but taking out the top 20 genes that are driving this discrimination, we can see that there is some differentiation between a disease fate. So we see some highly expressed genes based off of a disease outcome, an unfavorable one. And then we see some that are downregulated prior to disease exposure, and it will have just a pathogen challenge. So overall, these patterns allow us, these machine learning, allow us for 40 different biomarker candidates to be identified within this study and the shared species. And this allows us to find biomarkers that are both relevant to disease outcome, but also the prevalence within these species. And this application of biomarkers is extremely important to us going forward, as it will allow us to have continued coral monitoring and intervention as biomarkers may provide ecologically or immunology relevant biomarkers to curb these disease outcomes. I'd just like to thank Midlars Lab members who have worked on these experiments for allowing this computational experiment to occur. Thank you.